Hello everyone from Strasbourg. My name is Anaïs Buissot and I work in the Certification of Substance department here at the EDQM. And today it's with pleasure that I'm going to present you a general overview of the CEP procedure, its place in the regulatory system, and how it can also provide an advantage for manufacturers of active substance. In this presentation, we will be developing the following topics. First, we will look at the regulatory background of the certification procedure. Then we will compare the differences between the CEP and the active substance master file procedures. We're going to look at how the procedure works, how to apply for a CEP, and how the assessment takes place. Finally, we will go through some key figures and tools on how to keep updated with our activities. So let's begin with the background of the certification. And before starting, I would like to remind you the meaning of a CEP, which stands for Certificate of Suitability to the Monographs of the European Pharmacopoeia. The procedure was established in 1994 and was initially only applicable to pharmaceutical substance. It was then extended to products with a risk of transmissible spongiform encephalopathy, or TSE, it's easier, and then to herbal drugs and herbal drug preparations. Now, regarding the legal background of CEP. The context of our work is based on EU directives, and specifically the Directive 2001/83/EC, which is the legal text describing the regulatory environment for marketing authorization procedures for medicinal products in the EU. According to this document, the monographs of the European Pharmacopoeia are legally binding, which means that these monographs need to be respected when submitting marketing authorization applications in Europe. EU legislation has recognized existence of CEPs, as you see here in the text highlighted in the blue box, which states that where the active substance is the subject of a monograph of the European Pharmacopoeia, the applicant can apply for a certificate of suitability that, where granted by the EDQM, shall be presented in the relevant section of the CTD module. It also states that those certificates can replace the relevant data of the corresponding section in marketing authorization application. Another important aspect of these legal texts is that it recognizes the fact that although the monograph of the European Pharmacopoeia are authoritative and legally binding, there may be cases where the specific monograph alone is not sufficient to control the quality of a specific source of substance, and more appropriate specification may be requested from the competent authorities. Now we are going to enter more into the details of the CEP procedure itself. So the governing document for the certification procedure and its general process is described in this resolution document, which you can readily access on the EDQM website as shown on the slide. This resolution document first details the scope of the CEP procedure. So for chemical or herbal substance, the procedure is opened for those covered by a monograph of the European Pharmacopoeia. And for these, a chemical or a herbal CEP can be granted. Transmissible spongiform encephalopathy or TSE certificates can be issued for any substance for which there is a risk of TSE, as described in the European Pharmacopoeia General Chapter 528. And in this instance, there is no need for the substance to be covered by a specific monograph, and so this will lead to a TSE CEP. When you see CEPs, also please remember that those do not replace certificates of analysis. Neither does it replace a QP declaration, and nor is it a GMP certificate. The CEP procedure is not restricted to Europe, and it is open to any manufacturer in the world, regardless of its geographical origin. Further, CEPs are recognized by all member states of the Council of Europe and European Union, and are also accepted in some non-EU countries, such as Canada and Australia. In terms of substances that are out of the scope of the CEP procedure, those substances that are not covered by a specific monograph are out of the scope in case of herbal or chemical CEPs. Also, substances that do not comply with the definition section of the monograph are out of scope. So, for example, if the monograph states that the substance must be obtained through a synthetic route, it is not possible to apply for a CEP if the substance prepared by a manufacturer is obtained through a fermentation process. This is because scientifically, we would expect for the impurity profile to be different, and therefore it's likely that the monograph of the active substance would not control the final substance adequately. Biologicals are also out of the scope of the CEP procedure, as well as tissue derivatives, blood derivatives, vaccines, and finished products. As mentioned previously, the CEP is intended to demonstrate that the quality of a given substance can be suitably controlled by the relevant European Pharmacopoeia monographs with additional tests if necessary. The main purpose of the CEP procedure is to perform assessments of the quality of active substance on the basis of the monographs of the European Pharmacopoeia. The CEP procedure also provides a platform through which we can identify the need to update European Pharmacopoeia monographs. This is facilitated by the fact that through the CEP assessment procedure, we can detect potential deficiencies of the European Pharmacopoeia monograph, which can then be communicated directly to the European Pharmacopoeia Department, EPD, of the EDQM, allowing for a fast and secure transfer of information. The CEP procedure also provides a centralized assessment, which allows to save time for manufacturers and for authorities. The assessment is done once through all of Europe and does not need to be performed for each country. And this applies for both initial submission and variations, thereby allowing for significant saving in times and resources. Intellectual property is protected in this procedure since the information provided by manufacturers are treated in a confidential manner, and all assessors involved sign a confidentiality agreement and a declaration of interests. So now that we have seen the context of the certification procedure, we will go further into the details by comparing the CEP and the ASMF procedures. So first of all, drug substance documentation is an integral part of a marketing authorization application. And in the EU Note for Guidance, summary of requirements for active substance in the quality part of the dossier, you can see that in Europe, manufacturers can choose amongst 3 options to submit data on the quality of an active substance. These options are the certificate of suitability, the active substance master file, or to provide full details of manufacture of the active substance in the marketing authorization application. The scientific data is basically the same for all those options. CPs are not mandatory, but they generally avoid any subsequent reassessment. So now let's see the differences between the CEP and the ASMF procedures. And one of the main differences lies in their purpose. The CEP is independent from a marketing authorization application, and it confirms that an active pharmaceutical ingredient complies with European Pharmacopoeia requirements, whilst the ASMF is submitted in the context of a specific marketing authorization application for medicinal products. This means that in the CEP procedure, the final use of the substance is unknown, but it also means that it is possible to apply for a CEP even if the manufacturer of the API does not yet have a customer for the substance. Now looking at the scope, as we mentioned earlier, the CEP is intended for existing substance. So if there is no European Pharmacopoeia monograph for a particular substance, it will not be possible to apply for a CEP. By contrast, ASMFs are applicable for both new active substance and existing substance. Regarding the content of the dossier, it is essentially the same for both the CEP and the ASMF procedure. However, an important difference is that in the CEP procedure, the dossier is submitted to the EDQM by the manufacturer, and there is no need to divide the module 3.2s part of the dossier into an applicant part and a restricted part. Whereas for the ASMF procedure, the applicant part is sent to the marketing authorization holder, and the full dossier is submitted to competent authorities. The CEP procedure is a centralized assessment, which means that there is only one single evaluation, and generally, there is no need for reassessment of this part of the dossier by competent authorities during marketing authorization application. In addition, it's important to note that the assessment reports are available to European competent authorities and also other competent authorities who have a confidentiality agreement in place with the EDQM, and this allows to limit duplication of work. On the other hand, in the ASMF procedure, there is an assessment by each national competent authority. The principles of evaluations and references used are pretty much identical for both procedures. They are risk-based and science-based. At the end of the CEP assessment procedure, the certificate of suitability, the CEP, is issued to the holder, who can then provide a copy to its customers, and the CEP can replace that part of the marketing authorization application. In the ASMF procedure, an approval is obtained and no formal document is released. In terms of life cycle management, both procedures are very similar. The dossier must be kept up to date and variations submitted accordingly and in due time. And when a revised CEP is issued, it must be submitted to the national competent authority. Finally, the ASMF can be used in all EU and European Economic Area member states and also the UK, Australia and Canada, whereas the CEP can be used in all 39 European Pharmacopoeia member states. This includes all the EU EEA member states, as well as other members of the Council of Europe, and also a growing number of other jurisdictions, such as the one listed on the slide, and this list is constantly evolving. As explained in the previous slides, principles of evaluation of CEPs and ASMF are similar. And therefore, it's important to bear in mind that in order to reduce duplication of work and to improve efficiency, cooperation and work sharing have been progressively put in place. For example, in the CEP procedure, when an application is received, the applicant is asked to provide an agreement that we share EDQM assessment reports with other competent authorities. So you can see on the slide with whom those reports can be shared with. And this is, of course, for their own interest, as it can speed up the process. On the other hand, the EDQM can also request ASMF report if it is available, and this also significantly speeds up the process of evaluation, and it also contributes to harmonization of assessment within Europe. This closes now the section on the comparison of ASMF and CEP. And now we will focus on how to apply for a CEP. So, what should be provided in a CEP application? First of all, an application form, which should contain information on the CEP holder and manufacturing sites involved in the manufacture after introduction of starting materials. This includes their names, their addresses, GPS coordinates, as well as EMA SPOR OMS organization and location ID. We also expect a number of declarations, such as the confirmation of manufacture according to presented dossier and good manufacturing practices, willingness to be inspected declaration, the use or the non-use of material of human or animal origin, commitment to provide samples if needed, etc. The application form is also where the applicant can declare if they wish a specific grade to be included on their certificate, for example, micronized or sterile grade, and also if you wish to claim for a retest period. You can see the fees listed on the slide. In the application form, the applicant is also asked to include details on the marketing history of the final substance and to include the details on accepted ASMFs after October 2012. As mentioned earlier, this may allow EDQM to request ASMF and to speed up the process. The CTD dossier should also include a quality overall summary. As a reminder, the QoS is a mandatory component of the dossier, which gives an overview of the control strategy. And since January 2024, we have a new template available on our website that you can access as shown on the slide. So the dossier should preferably be submitted in English, but the applicant could also submit it in French, since this is the other official language of the Council of Europe. The dossier should be built in accordance with the relevant policy documents listed on the slides. And you have also some new guidance for applicants wishing to include a sterile grade on their CEP. All of these documents are on the EDQM website for free, and applications should be submitted in eCTD format via the European CESP platform. And to use this platform, applicants should register at the Heads of Medicines Agency's website. So now we will see how the evaluation process works and the flow during the assessment. So when a CEP application is received, it first goes through a validation step to make sure that it is within the scope and suitable and ready for assessment. When this step is performed, the evaluation can start. The evaluation is usually done by 2 assessors, and they write an assessment report, which we call a report A. At the end of the evaluation, the assessors can conclude with either one of these possibilities. First of all, the dossier can be found compliant with requirements and the CEP is granted. Second possibility, it could be that there are information missing to confirm compliance with the requirements, and so a further round of assessment is needed. And in this case, a request for additional information is sent. And when the information requested is received, it goes through the same step again. Rarely, it can also be found that the dossier is too deficient and the procedure is closed. During the assessment, the assessors can identify a need for a GMP inspection. For example, if there are doubts regarding GMP compliance. And in this instance, the assessors can prepare what we call a report C, which is forwarded to the EDQM inspection team. The assessors can also identify a need to improve and revise the monograph, for example, when there are additional impurities not listed on the transparency statement of the monograph, so the list of impurities which is on the specific monograph. In this instance, the assessors can prepare a report B asking for the inclusion of this impurity to the monograph. The report B is forwarded directly to the European Pharmacopoeia Department, the EPD, for the treatment and the potential revision of the European Pharmacopoeia monograph. The CEP needs to be renewed after 5 years. However, now with the concomitant existence of CEP 2.0, hybrid CEPs and old CEPs, this does not necessarily mean that you will receive a revised CEP. An important note regarding companies' response in additional rounds of assessments. So after an assessment, as mentioned before, the EDQM may send to the company a request for either additional information, for clarification, or for dossier update. And so according to our policy, the company should ensure that their responses only contain information requested by EDQM and not additional changes which are not related to the questions asked by EDQM and that may have been omitted in the initial round. There are some exceptions to that, which are administrative changes, which can be submitted in the course of a response to additional information, dossier update after a revision of the monograph, or submission of additional stability data to support longer retest period. So how long does the process take for a new CEP application? So the EDQM timeline for the initial assessment is 115 working days, which is around 5 to 6 months. And the outcome of this initial assessment will define the applicant deadlines. So if significant information is required, the applicant will have 180 calendar days, so 6 months, to address the points raised in the request for additional information. And upon reception of the data, the EDQM timeline will be of 92 working days, so around 4 months. The applicant can also have only minor issues to address, in which case they will have 30 calendar days, so a month, to respond, and EDQM will have also about a month to finalize the second round. And if necessary, this goes on for a third round. I put here the link to the policy document detailing all the deadlines depending on whether your application is a new application as shown on the slide, or a minor revision, or a major revision, or a renewal, or a sister file, etc. Please also note that, as published on our website on the monthly report, we're still experiencing some delays for new dossiers, so for new applications, and we apologize, and we're really doing our best to reduce those delays. So who performs the evaluation? The evaluation procedure is monitored by a steering committee. The steering committee is in charge of taking decisions and giving advice on regulatory issues associated with the implementation of the procedure. And it's also the steering committee who appoints the assessors, so the ones performing the evaluations. So as I mentioned earlier, in the evaluation of a new chemical CEP application, for example, the evaluation is done by 2 assessors, and these assessors can either be EDQM staff members or experts from national competent authorities. They work together during assessments, and the dossiers are allocated according to their expertise, whether that be chemical evaluation, TSE risk, or toxicological assessment, for example. The experts come regularly to the EDQM for these evaluation sessions. It's more or less every 2 weeks. There are about 100 assessors from authorities from 25 countries, including Canada. We now also have remote sessions, mainly since COVID. So as you can see, this is a great and successful example of international cooperation. So we'll mention briefly the sister file procedure here. So through the sister file procedure, a company which has been already granted a certificate of suitability may wish to apply for another CEP for the same active substance. For example, when the company wishes to have separate CEPs for different conditions of preparation or qualities. So for example, when they want to cover an alternative manufacturing process, manufacturing site, or an alternative grade. The sister file procedure is really intended to facilitate the submission to the company, so the submission of similar dossiers within the certification procedure, and to allow applicants benefit from a fast-track procedure since it's treated within 2 months, and to have harmonized assessment. There is some guidance available on our website as referenced on the slide. So now we will look at some key figures. So since the procedure was established, close to 9,000 CP applications have been received for nearly 1,500 different substances. There is currently more than 6,500 valid CEPs from about 1,500 manufacturers from all over the world, mainly from Asia and Europe, as you can see. So the CEP procedure, as mentioned before, is not mandatory, but it is widely used. Those manufacturers have voluntarily applied for CEPs as it is widely recognized in the regulatory environment. I also strongly encourage you to look at our website. So on our website, you can have access to our monthly activity report, which includes details on how many submissions we receive every month, the numbers of CEP granted, the average time for treatment, the number of inspections that have been carried out, etc. You can also access to a lot of information on our website. For example, the guidelines to the procedure. You can also find the certification database. This allows you to check the status of CEPs. You have also a lot of information of the CEP 2.0. You can also find the links to the FAQ and to the help desk, as well as some e-learning. So again, I strongly encourage you to have a look at the website. Finally, in terms of communication with the EDQM, so we have developed FAQs. So should you have any question that is not on the FAQ, you can also ask general question on the EDQM help desk. So you can find a link on our website as shown previously. And for a specific question regarding specific application or submission, please send your queries via the email address included in the EDQM communication. Finally, there is also the possibility for a technical advice procedure, which allows you to meet with EDQM staff to get advice on a specific or a future application to ensure that it meets requirements. So thank you very much for your attention. I hope that this presentation has given you a general overview of the CEP procedure and that you found the presentation useful. And now I will hand over to my colleague, Ekaterina, who will present the topic use of a CEP. Thank you very much.
So, hello, ladies and gentlemen. Thank you, Anais, for your presentation. My name is Ekaterina Nagdiev, and I'm a supervisor in the revision team in the Certification of Substances department. And I will continue with you now on the second part of the presentation, which is related to the use of the CEP and the informations which are stated on the CEP. So in particular, we will see in this part of the presentation the aim and scope of EDQM public document, How to Read CEP, which is currently under revision, but it will be published soon. We will see how to interpret the information which is stated on the CEP, and we will see practical examples for chemical CEPs. We will highlight the most important changes following the implementation of CEP 2.0, and we will see how to use CEP in the marketing authorization applications. So EDQM public document 1531, How to Read CEP, describes the information which is stated on the certificates of suitability and clarifies how to read and how to understand this information by the industry and competent authorities. It does not cover the use of CEP in the context of marketing authorization application, where the CEP is used to replace the quality part of the CEP. And for this, there is a specific question and answer document published by EMA, which describes how to use and how to include CEP in the marketing authorization applications. This document does not include any new information or any new instructions. It's rather a compilation of all of the existing instructions and policy documents. And these documents should be read in conjunction with other policies and guidelines of CEP procedure. So as you probably know, it exists different types of CEP. Chemical CEP or herbal CEP certifies that the substance is suitably suitably controlled by EP monograph and any supplementary test deemed necessary in line with ICH EMA guidelines. Also, the TEC certificates certifies that the substance complies with the general chapter 5 to 8 on the minimizing of TEC risk. This TEC does not certify that the substance is suitably controlled by specific EP monograph. As Anais previously said, CEP does not replace the certificate of analysis, it does not replace QP declaration, and CEP is not a GMP certificate. So now we will see how to read the information which is stated on each type of CEP. Following the switch to CEP 2.0 format, which took place in September 2023, three different formats of CEP have been introduced. CEP 2.0, which is new format, hybrid CEP, and old format. So now there is coexistence of these three formats of CEPs, and the presentation is not the same. So on this slide, you can see the differences between these different CEP formats. For example, CEP 2.0 and hybrid CEP are electronic documents with electronic signature, whereas the old CEP was a paper document with a wet signature. The numbering also changed. There is now the reference to the renewable procedure. We kept the renewable procedure in CEP for CEP applications. However, it does not result in the new renewed CEP if the content of CEP didn't change. So for all CEPs, they following the renewal procedure, they switch to the CEP 2.0. If the CEP is in hybrid format, so the applicants, you may ask to switch to CEP 2.0, and this should be stated on the cover letter. However, if there are no changes to the content of CEP, CEP remains in hybrid format. So on CEP 2.0 and hybrid SEPs, now we state OMS, SPOR OMS IDs, log and org ID, whereas it was not the case for the old CEP format. Also, the technical information on the CEP has changed. In the context of CEP 2.0, now the specification, the approved specification, and the additional test procedures which are necessary to control the final substance are annexed to the CEP. Whereas for hybrid and OCEP, additional limits are stated on the CEP itself, and the additional tests are appended. So for CEP 2.0 and hybrid CEP, there is no expiry date, 5 years, whereas for the old CEP, there was expiry date of 5 years and before renewal, and after renewal, there is no longer expiry date. So also in terms of your authorization to use the CEP, which we will see in the next slide, it changed. For CEP 2.0 and hybrid CEP, you provide a letter of access to your customer. For old CEPs, box of access was used for this. And as I already told, this change was implemented in September 2023. So all new certificates which are granted from September 2023 are in hybrid or format or CP 2.0 format. So by the letter of access or the box of access, the CP holder authorizes its customers to use CP in support of the marketing authorization application for a particular product. So for new CP and for CP 2.0 and hybrid CP, this is done by filling a letter of access, which is provided together with a copy of CP received from the EDQM. For the old CP, it was by completion of a box of access, which should be suitably completed on a copy of the CP received in old format from the EDQM. And as Anais already told, CP is accepted in all Pharmacopoeia Convention member states, plus some other countries, and this list increases. It's Canada, Australia, Singapore, South Africa, etc. It's important to emphasize that the communication between CP holder and the drug product manufacturer is the key. The drug product manufacturers and marketing authorization holders are ultimately responsible for the quality of the APIs that they use to manufacture their finished products, and they are legally obliged to get the necessary information to take this responsibility. On their turn, API manufacturers should be supportive to their customers and share the needed information. And the responsibilities of the CP holders towards their customers are described in a specific EDQM policy document 2157. EDQM continues working with stakeholders to promote better sharing on information in the benefit of everyone. So now we will see most specifically the informations which are stated on different types of the CPs, and we will start with the chemical CP. A CP can cover specific physical-chemical characteristic of a substance, for example, specific polymorphic form or particle size distribution, which are related to their substance, or it can also be done for sterility. And these additional characteristics are covered by subtitle, which is mentioned on the CEP. Subtitle is optional. It should also be, yes, it should also be highlighted. So that subtitle can also be used to distinguish CP applications for the same substance from the same holder if they use alternative manufacturing process, for example, process B, or if the substance is produced at the alternative manufacturing site, for example, site X. In addition, a subtitle is also used to reflect on the CEP the requirements of the labeling section of the monograph. For example, when for the presence of antioxidants for the API mixes. So the grade is mentioned on the CEP as a subtitle only if it is requested by the CEP holder clearly in the application form, and if it is accepted by the EDQM during the CEP evaluation procedure in line with the EDQM document 041, content of the dossier for chemical purity and microbiological quality. So we will see the examples for subtitle. If a subtitle is requested and accepted by the EDQM, the corresponding quality attributes are included in the specification appended to the CEP in case of CEP 2.0 or mentioned on the CEP hybrid or old SEPs, and the analytical methods which are used to control the specific quality attributes are annexed to CEP. CEP may mention more than one grade. In case if the substance of these different grades, for example, micronized and non-micronized, has the same impurity profile and the same specification is applied, not taken into account these tests which distinguish different grades. If the impurity profile is different, separate CEP applications should be submitted and separate SEPs will be issued. For example, for micronized, non-micronized grade, data related to determination of particle size is assessed by the EDQM. In case of polymorphic form, the data related to the elucidation of specific polymorphic form is assessed by the EDQM. So as I told, subtitle is not mandatory, it's optional. And when there is no subtitle on the CP, this means that a particular grade has not been requested, has not been included in the CP dossier, or the substance, the CP holder did not apply for a specific subtitle to be mentioned on the CP. As you know, manufacturing sites are mentioned on the CP with their rows. So names and addresses are mentioned for CP holders, intermediate manufacturers, and the final substance manufacturers. The manufacturers of starting materials are not mentioned on the CP. Moreover, additional sites which are involved in additional treatment to receive a specific grade are also mentioned on the CP, and this is the change following the implementation of CP 2.0, and this might be the sites for physical treatment, micronization sites, sterilization sites. Now with the switch to CP 0.0, SPOR, OMS log and org IDs are mandatory for all sites at CP, and please pay attention that the information is in line with the information stated in EMA OMS database. Regarding the production sites are mentioned in the Annex 1 of the CP. Regarding the intermediate manufacturers, the names of the intermediates which are concerned are not specified on the CP in the annex. And the CP does not distinguish which manufacturer produces which intermediates if more than one intermediate is involved, for example. So it is the responsibility of CP holder to communicate with the CP users and to share the information which needs to be included in the marketing authorization application regarding the responsibilities of intermediate manufacturers. As you know, in the context of CP 2.0, now the specification of the substance is appended to the CP. Specification should be based on the corresponding EP monograph, ICH and EMA guidelines, and the compliance of these tests are checked and approved during CP evaluation procedure. Specification should include the limits of the related substances and other limits which are foreseen by EP monograph plus additional tests and limits which are required for this might be additional in-house process-related impurities which are not mentioned in the transparency statement of the monograph if needed. So the specification table should include quality attributes, acceptance criteria, and also the reference to the test procedures which are used to the control. The final substance should be mentioned, and the reference should be given to EP test, pharmacopoeial test, or in-house test. Specification may also include the information on skip testing if the skip testing is foreseen by the corresponding ICH or EMA guideline. For example, for elemental impurities in line with ICH Q3D, for mutagenic impurities in line with ICH M7, for nitrosamines in line with EMA question and answer document on nitrosamine impurities. However, it should be mentioned that skip testing has to be approved by the relevant authorities reviewing the marketing application for the medicinal products where it is included. Regarding the test procedures, and this applies to all types of CEPs, 2.0 hybrid and all CEPs. Alternative test procedures to those which are described in EP monograph, for example, developed in-house or taken from other pharmacopoeias, may be used, provided that they are at least equivalent to the EP monograph. So in this case, cross-validation data should be demonstrated, should be provided to demonstrate that the methods are at least equivalent, and this information is assessed by the EDQM. And in the event of doubt on dispute, please be reminded that the text of the monograph remains authoritative. When EP monograph is demonstrated to be suitable to control the quality of the substance, the in-house tests is not appended to the CEP. For the quality attributes, I'm sorry, I switched. Yeah, for the quality attributes not covered by the EP monograph, but which are needed to control the quality of the substance, company's test procedure, in-house test procedures are appended to the CEP. For example, the GC methods for in-house GC methods for residual solvents. This might be the tests for elemental impurities, all the additional tests. Yeah. For hybrid and old steps, the specification is not appended to the CEP. In this case, the test required in addition to the corresponding test of the monograph are reported on the CEPs, and additional related substances can be, in some cases should be listed on the CEP. I can share with you the two most common scenarios for additional related substances which are mentioned on the CEP. So if an impurity is present in the final substance above the identification threshold of the General Monograph 2034 Substances for Pharmaceutical Use, these impurities are stated to the CEP and they should be specified with the specified limits. So if they can be controlled by the test for the related substances described in the monograph, the test procedure is not appended to the CEP. For example, in this example, impurity A is present higher than 0.10, the identification threshold, and it is limited at 0.15. It's a specified identified impurities by the test of the monograph. If the impurities are present in the substance above the reporting threshold, and the EP monograph is not suitable to control these impurities, they are controlled by a validated in-house methods. So the limits of these impurities are stated on the CEP, and in this case, the in-house method is annexed to the CEP. For example, in this case, a specific impurity which is limited on the CEP at 0.10, and in the Annex 2 here, the in-house method for related substances is annexed to the CEP. So in some cases, which is a rare case, when the current monograph does not include a limit for unspecified impurities, such limit has to be introduced in the specification and included on the SEP, in line with the EP General Monograph 2034. This case is a rare case. This can be still the case for some old monographs. They are progressively revised, but there are still this scenario for some files. For CP 2.0, the statements on the impurities are not stated on the CEP, but the limits for unspecified impurities are transparent from the specification which is appended to the CEP, and this limit is included in any case in the specification. Then, in some cases, EP monograph may describe still a non-quantitative method, for example, TLC method for related substances. In this case, the manufacturer should replace this non-quantitative test by a quantitative validated in-house method, and the in-house method in this case is appended to the CEP. TLC method can ever be acceptable for control of one, for example, specified impurity, but it cannot cover all the tests for related substances. And in this case, the statement on the CEP will be as follows. So the test for related substances by the thin layer chromatography described of the monograph is replaced by the test by liquid chromatography in-house test, and the limits are stated on the CEP. So about the mutagenic impurities. So mutagenic impurity is limited on the CEP or it is controlled in the specification which is appended to the CEP if it is present or can potentially be present in the substance. The limit proposed by the CEP holders is assessed and accepted by the EDQM in line with ICH M7 requirements. If the method of the monograph is not suitable to control this impurity, in this case, the in-house method, specific in-house method to control for this impurity is annexed to the CEP. But what does it mean if non-mutagenic impurities are limited on the CEP? So this means that either there are no potential mutagenic impurities which can be formed or introduced in the specific route of synthesis applied by the API manufacturer, or there are potential mutagenic impurities, but the control strategy put in place by the manufacturer enables not including a limit in the final substance specification. So the impurity may be controlled by option 3, 2, or 4. And after the assessment, when it's concluded that option 1 is not needed for a specific impurity. Or in some cases, mutagenic impurities can be controlled by the monograph. Now we will speak about the residual solvents, and we will start with the case for old and hybrid CEPs. So which solvents are mentioned on the CEP? On the CEP are controlled the solvents which are likely to be present in the final substance and those which are used in the last steps of the manufacturing process. So this is covered by the Note for Guidance for Residual Solvents, EMA Note for Guidance for Residual Solvents, and this decision tree will explain you the approach that is applied. So the solvents, the first question is solvent is used in the last step of the manufacturing process? If yes, it should be controlled in the final substance. If the a solvent is not used in the last manufacturing process, a carryover data should be included in the CEP application for all solvents which are used in the manufacture of the final substance. And if a solvent is detected at levels above 10% of ICH option 1 limit, or above 30% for class 1 solvents, in line with Note for Guidance for residual solvents, the control should be implemented and the control will be stated on the CEP. On the other hand, if the solvents are detected below 10% of ICH limit for class 2, 3 solvents, or below 30% for class 1 solvents, the control is not considered to be mandatory, and the solvents, it is considered that the solvents are demonstrated absence in the final substance and the test is not mentioned on the CEP. So in any case, limits proposed by the manufacturers are assessed and should be accepted by the EDQM. The most common 3 scenarios on the limiting the solvents on the CEP. So the most common scenario when the solvents are limited on the CEP in line with option 1 limit of ICH Q3C. Sometimes title limits can be proposed by the companies and the specification limits which are stated in the internal specifications are stated on the CEP. This means title limits. Exceptionally, in some cases, higher limits than option 1 are acceptable if it is suitably justified, for example, applying option 2, and in this case, this is made clear and transparent on the CEP. The test for Lawson drying. In some cases, you will see on the CEP that some solvents are controlled by the test for Lawson drying, and this applies for class 3 solvents. So in case if Lawson drying test is included in the monograph with a limit at not more than 0.5%, and class 3 solvents are likely to be present in the substance, you will see the following statement on the CEP that in the last steps of the synthesis, isopropanol, for example, class 3 solvent is used as solvent, and the residual content is limited by the test for Lawson drying described in the monograph with a limit at not more than 0.5. Or, for example, if the Lawson drying test is not included in the individual monograph, but the manufacturer decides to use the general test for loss on drying 2 to 32 to control water and related substances, the following statement is mentioned on the CEP. In the last steps of synthesis, water and acetone are used as solvents. Their residual content is limited by the test for loss on drying 2 to 32 of the European Pharmacopoeia with a limit at not more than 0.5%. So now we will see the specific example on how the limits may be stated in the specification, internal specification, and how they will be stated on the old and hybrid CP. I again draw your attention that it's not for CP 2.0, we will see later. So in the applicant's specification, there is a test for loss on drying at 0.5%, and there is the test for residual solvents. In this case, you will see for example, the whole battery of solvents which is used in the process. However, on CP, you may see only the in-house test for methanol and the statement on the use of acetone in the solvent in the last steps of the process, and its control by the test for loss on drying. Why? There is a difference between in-house specification and the information mentioned on the CEP. So in this case, we can understand and interpret the information that acetone and methanol are used in the last steps of the process, in the last purification steps. All other solvents are used early in the process, and they were found below 10% of ICH Q3C option 1 limit in the substance, though these tests are not mandatory. And in addition, acetone is class 3 solvent, and when there is a test for loss on drying in the monograph, it can be controlled by the test for loss on drying of the monograph. Following the implementation of CP 2.0, the limits for residual solvents are stated in the specification, which is appended to the CEP. So in this case, we take the specification provided by the companies, and the specification, of course, after assessment is annexed to the CEP. This means that if all solvents used in the process are limited in the specification, these controls are appended to the CEP as part of the specification. So this is a difference between old, hybrid, and CEP 2.0. If class 3 solvents are used in the last steps of the process and controlled by the test for loss on drying of the monograph or by the general chapter on loss on drying, a corresponding statement is still mentioned on the CEP. In some cases, water is mentioned on the CEP. So water is mentioned on the CEP if it is used in the last steps of the manufacturing process, and it is likely to be present in the final substance. With the implementation of CEP 2.0, now the quality of water additionally is mentioned on the CEP. So, and please pay attention that the quality of water mentioned on the CEP should be in line with the EMA guideline on the quality of water for pharmaceutical use. So this means that the grade of water which will be mentioned on the CEP is potable water, purified water, or water for injection. And when it's clear in the file from the beginning which type of water is used, and the specifications are in line with the water which is declared to be used in the process, this also simplifies the assessment process and avoids any additional questions. About the elemental impurities, as you know, ICH Q3D guideline on elemental impurities has been applied for medicinal products for human use from September 2016. From January 2021, the risk assessment regarding the elemental impurities also performed for veterinary medicinal products. In the CEP procedure, the document which describes how to implement the policy on elemental impurities is described in the EDQM public document 1623, which was revised in April 2021. So it should be mentioned that ICH Q3C applies for finished product, not for APIs, and the EDQM does not take decision on the compliance with ICH Q3D. However, the CP provides transparency on the use of elemental impurities to be considered by the manufacturers of finished products in the context of marketing authorization applications. You may see the following statements on the CP. A risk management summary for elemental impurities has been provided. It applies for old and hybrid CEPs, or the section miscellaneous information includes the risk management summary for elemental impurities. This means that in the CEP application, the risk assessment and risk management summary is provided for elemental impurities by the CEP holder, and its summary is annexed to the CEP in line with EDQM public document for elemental impurities, with the necessary information on the presence and absence of elemental impurities in the final substance and their levels of elemental impurities. When the risk management summary is not provided, the CEP at least is transparent on the introduction of elemental impurities, not about their levels, absence, presence, but on the introduction. And you may see the following statements. No elemental impurities classified in ICH Q3D is intentionally introduced in the production of the substance, or the following elemental impurities classified in ICH Q3D are intentionally introduced. In this case, it's lead and palladium. In some cases, the applicant might have set a limit for specific elemental impurity in the final substance. In this case, the limit will be stated in the CEP and the test procedure will be annexed. So on this slide, you see the layout of the risk management summary for elemental impurities, and which is provided as Annex 1 to the EDQM policy document 1623. And please pay attention to complete it with the necessary informations on the route of synthesis, on the elements which are intentionally introduced in the manufacture of the substance, of the final substance from the introduction of the starting material, and the conclusion column should be properly completed with the absence, in line with the definition of absence which is mentioned in the footnote. So it's less than 30% of ICH option 1 limit. You may include also the levels found, the maximum levels found of the elemental impurities in the substance if option 1, for example, is not applied. Option 2 can be applied when justified. In this case, it should be transparent on the maximum daily dose which was used to justify option 2A. You also may state that no risk is identified for several impurities. So omission of monograph test is possible in some cases when it is demonstrated that specific test described in EP monograph is not necessary for a specific named compound because this impurity solvent cannot be present with the applied route of synthesis, or if it's not used. So omission is acceptable for specific tests for one or few impurities. However, of course, we cannot apply it for the test for related substances. So for CEP 2.0, the omission is transparent from the specification which is appended to the CEP when a test is not stated in the specification and it was assessed and approved by the EDQM. About the microbiological quality. So in general, if it's not foreseen by a specific EP monograph, microbiological control should not be part of the specification proposed in a CEP dossier. Microbiological quality is covered by General Monograph 2034 substances for pharmaceutical use, and it should be addressed considering the final use of the substance in the finished medicinal product. And it should be evaluated by the national competent authority who receives the CEP in the marketing authorization application. Container closure system is also described on the CEP, even if in some cases no retest period is requested by the CEP holder, and it includes immediate packaging, primary packaging, and outer, secondary, or tertiary packaging. You will see some examples on these slides. For example, the substance is packed in the double polyethylene bag, in a triple laminated bag with a different um, definition of each layer, placed in the polyethylene container. Or substance is packed under nitrogen atmosphere in epoxyphenolic resin-lined iron drum. Sometimes alternative packaging may be also included and stated on the CEP. For example, the substance is packed either in polyethylene bag, in a polyester aluminum composite bag, or in a double polyethylene bags, outer bag, placed in either fiber or paper drum. Retest period is optional, but it is highly recommended. So the CEP statements on the retest period reflects the fact that the substance is stable during a certain period, 36, 48, 60 months mentioned on the CEP, in a specific packaging material mentioned already on the CEP, on the long-term conditions, 25 or 30 degree, in line with the relevant ICH guideline. Short-term excursions are covered by additional testing at the accelerated test conditions. For example, you see here an example. The retest period of the substance is 36 months if stored in amber glass bottles with polybutylene screw caps placed in the polyethylene bags. So different retest periods and storage conditions can be proposed within one CEP applications. For example, different container closure system, as I previously told, or different climatic zones. What does it mean if a CEP does not include a retest period? So this means that the retest period has not been requested by the CEP holder, and in this case, stability data are not assessed, or might be requested by the CEP holder. However, stability data presented in the dossier did not allow granting a retest period. For example, in case if there is insufficient data, some out of specs, or the storage conditions are not in line with the respective stability guidelines. In any case, stability data should be evaluated during the assessment of the marketing authorization dossier. Alternatively, when there is no retest period, the finished product manufacturer should demonstrate that the substance complies with EP and additional CEP requirements immediately before its used in the manufacture of finished product. So in most of the cases, the specific storage conditions are not mentioned on the CEP, and this means that the substance is stable under climatic zones for conditions for zone 1 and 2, and in long-term and accelerated conditions. But in some cases, CEP indicates specific storage condition. And in these cases, they are needed to ensure the stability of the substance in the described container closure system, or the CEP holder is applying stricter storage conditions than those recommended by the ACH guidelines, which is now also accepted. For example, the retest period of the substance is 36 months if stored in the temperature between 2 and 8 degrees in the described packaging material. But in any case, the retest period is supported by stability data obtained in the appropriate conditions. Also, the CEP contains specific statements on the use or non-use of materials of human or animal origin, and CEP applicants have to provide a declaration on whether or not they use materials of human or animal origins in the manufacture of their substance. So in this decision tree, you will see that if material of human or animal origin is not used in the manufacture of the substance, This results in a chemical CEP, and a statement on non-use of material of human or animal origin in the production of the substance is stated on the CEP. On the other hand, if material of human or animal origin is used, we should ask ourselves a second question: if there is a TEC risk related to the use of this material. If there is a TEC risk with the use of this material, this results in a double CP chemical and TEC, and the TEC aspects are evaluated by the specific dedicated assessment. If there is no TEC risk in connection to the use of this material, the assessment results in the chemical CEP, and the following statements can be mentioned on the CEP. So in EDQM may assess the risk of viral contamination, and if there is no risk, we state that material of human or animal origin is used in the production of the substance, and the process includes rigorous viral inactivation steps, which minimizes the risk of contamination. When the assessment is not possible at the EDQM, so it is stated that the viral safety has to be assessed in the context of medicinal product containing this substance. Some pharmacopoeial monographs contain production sections, which provide specific instructions to the manufacturers about particular aspects of the manufacturing process. For example, source materials, in-process testing, or testing to be carried out by the manufacturer prior to release. So not all these production section statements can be verified during the CP procedure. If they are verified and assessed, nothing is mentioned to the CEP. This means that these aspects have been assessed by the EDQM, and no further action is needed during the assessment of the marketing authorization dossier. If these aspects could not be verified during the CEP evaluation procedure, a specific statement is mentioned on the CEP, which states that the information on the production section is to be addressed during the assessment of the marketing authorization application. So for sterile substances, a sterile CEP does not exist on its own. It's always combined with a chemical CEP or with a double CEP. And in any case, the step includes the typical statement that we previously seen for each type of CEP. The European system requires that sterilization data should be included in the marketing authorization application, even if a CEP for sterile substance is submitted. So in any case, the data on sterilization should be included in the marketing authorization application for finished product. So for sterile substances, there is a specific subtitle on the CEP sterile. Sterilization method is mentioned on the CEP, and the statement regarding compliance with the test for sterility of the monograph on all CEPs is still mentioned, and the statement on the sterilization process, that the sterilization process has been assessed and accepted by the EDQM. Sorry. So the following statements you can see on the CEP. The substance is sterile and comply with the test for sterility of the European pharmacopoeia, and the method used for sterilization is sterile filtration. Sterilization has been assessed and approved. Now on the CP 2.0, it's mentioned that the substance is sterile and is produced by sterile filtration. So on the TEC certificates, you may see subtitle, for example, for the manufacturing process of gelatin. Also mentioned countries of origin of the source materials, name of nature of animal tissues used in the manufacture, and the manufacturing process applied, if relevant for the safety of the product, for example, for gelatin. So in this case, the CEP certificate does not certify that the particular source of the substance is compliant with the corresponding EP monograph for that substance. It certifies that the substance is TEC free. So it complies with the general monograph 1480 in 83 on the products with a risk of transmitting of animal spongiform encephalopathy. For herbal CEPs, for extracts, drug extract ratio calculated on the genuine extract without excipients is stated on the CEP. Residual solvents with acceptance criteria and control methods for them, for those which are used in the last step, extraction solvents, and the information of excipients used with the name and percentage of the excipients. And for all herbals CEPs, the packaging materials is described on the CEP as for all types of the CEP, release period if requested by the applicant, and the use or non-use of material of human or animal origin. So, as I previously said, the use of CEP in the marketing authorization applications in EU are covered by a specific EMA question and answer document on how to use CEP in the context of marketing authorization applications or variation. So please, yes, this document is available on EMA database and provide you with all necessary guidance on how to include CEP in the marketing authorization applications. So SEPs are accepted in countries also outside Europe at the discretion of the authorities of those countries. So those authorities decide on the scope of the acceptance of CEP and the conditions that may apply. So in some cases, they may require some additional information, for example, provision of DMF or other documents. And the applicant has to are encouraged to verify the acceptability and conditions associated with the use of the CEP before they submit their file. So that's all from me, and I hope that it was interesting and useful for you. If you have questions, please don't hesitate, and we will be happy to reply to your questions.
Hello again. So I see that we have received a number of questions. Sorry, I had a little bit of echo. So I will go through some of the questions that we have received. First of all, so please let us know where the CEP certificate can be placed in the CTD module during the application. So for this, you can refer to, so there is an EMA question and answers on how to use a CEP. Here you can find many information for both marketing authorization application and variations. And for where it can be placed in the CTD, so it states that there is a copy of the most recent version of the certificate with the declaration of access to the marketing authorization holder of or applicant, or a letter of access for the CEP 2.0 duly filled and signed. That should be included in the section 3.2R or the VNIS part 1. So again, I would strongly suggest to check out this EMA question and answers on how to use a CEP in a MAA or marketing authorization variation. So either an official list of countries with whom we share assessment reports. So yes, there is one. If you look on our EDQM website, on the general website, there is a certification section, and you can check out in the background and legal framework, and you will have a link to so the list of authorities and organization which may have access to assessment reports, and there are also inspection reports. So that will bring you to a table which lists countries, and it explains if we share either the assessment reports or the inspection reports or both. So to give you with well, I can briefly give you the list. This includes the ANVISA, so Brazil, Canada, Singapore, Israel, also South Korea, Japan. Then for Switzerland, we have also the WHO, which is in Switzerland, Saudi Arabia, South Africa, Taiwan, Australia, and US FDA. This list is constantly evolving, so if you want to make sure you have the last version, you can check this out on our website. So we also received a question regarding GMP inspections. So at which stage a GMP inspection can be initiated? So during the presentation, we mentioned one case, which is when going through the assessment of the dossier, if there is a suspicion of non-compliance, GMP non-compliance, we can draft a specific report, which is then forwarded to the EDQM inspection team. And this is one of the cases where it could trigger a GMP inspection. But what I wanted to highlight is that this Friday, on the 13th of December, I would really strongly encourage you to attend to the module 9, which is dedicated to the EDQM inspection program. So there is a live webinar on Friday, and so that will be the perfect place to know and learn more about the GMP inspection and the EDQM inspection program. So there is also a document regarding the declarations to be, questions regarding the declaration to be provided in the application form. So in the Annex 6 letter of commitment to provide samples upon request by the EDQM, is this required to be signed only by the API manufacturer or also the intermediate manufacturers if there are some and if they are different than the API manufacturers? So no, in this case, there would be no need to provide the letter of commitment to provide sample. This declaration is dedicated to the API manufacturer. And on the application form, if you have intermediate manufacturers in your process which are different than the final substance manufacturer, when a declaration is needed from the intermediate manufacturer on the application form, normally you can see it concerns those declarations where it is stated in the application form for it to be provided for each manufacturing site. So we also have a question on fast track procedures. What are the conditions applied to and the timeline for completion? So this specific case was discussed when mentioning the sister file procedure. So an example would be that you as a company already hold a CEP for an active substance, but now you would like to have another CEP for the same active substance. This is important. But for example, this time it's obtained via an alternative process. So let's say that in the last step of the process, a different solvent is used, or that the alternative process also leads to a specific polymorphic form. So those type of case, you have a guideline which details when you can cover an application in the frame of a sister file procedure. So then in this case, you can apply for a new dossier sister file, which is different in terms of timelines compared to the just new dossier application. So instead of the standard 5 to 6 months for the new dossier application, here in the sister file, you will benefit from a fast track procedure, meaning that the assessment of the first round will be 2 months. In terms of the timeline for completion, so you have this guideline, so available on our website, management of application, for new certificates of suitability, requests for revision or renewal of certificates of suitability, and application using the sister file procedures. So in the presentation earlier, I only showed this table for new dossier application with the timeline, but under the guideline I just mentioned, you will find the full table, which would include the timelines for new dossier application, sister file, minor revision, major revision, monograph revision, and renewal. So first, the question was dedicated specifically to the sister file. So you have 2 months for the initial assessment. Then the company will get a month to reply to the request for additional information. And then when we receive the reply back, we would have a month to treat the request for additional information. So, okay, we received a question which is asking what are the main reasons EDQM will not accept a grade in the CEP subtitle. So I'm not really sure from which part of the presentation this question was raised, but I think there might have been a misunderstanding. So I just want to mention that EDQM does accept grades in CEP applications. So an example would be a micronized grade or a sterile grade or polymorphic form. So I just wanted to mention that, but if you have some more question, we will repeat this at the end of the webinar. But if you have a more specific question, you can send us, you can check out either the FAQ or go to or raise a question through the help desk if it's a general question, or if it's for a specific question related to your application, you can also send us an email through the email communicated in our correspondence. Okay, I will just go through 1 or 2 more before giving the floor to Ekaterina. So you stated that the validity of CEP 2.0 is no longer mentioned, but is 5-year validity be unfaired? So yes, even with CEP 2.0, when the CEP is issued, it has a validity of 5 years. And if you go on our certification database, so this is again on our website, where you can check out the status of CEPs, whether they are valid, suspended, withdrawn. Even with CEP 2.0, you would have the issue date and you have a section called renewed date. And so you will see here the validity, which will be in 5 years. And then around the 5 years after first issuance, then you should submit a renewal application for this CEP to be renewed. Okay, I think I will now hand over to Ekaterina.
Yes, so now I will also reply to some questions which I received. It's true that we received many questions, so I will try to go one by one. So the first question that I see, is it mandatory to mention storage conditions and retest periods? So the storage conditions are stated as part of the statement on retest period if the retest period is requested and it is granted. So it is not mandatory, but if you ask for retest period, it will be mentioned. On the other hand, the packaging description is mandatory. So despite the fact retest period is mentioned or not, the packaging description is stated on the CEP. However, the addition of retest period is encouraged because it is if possible, because I think that it is beneficial for you as the final final substance manufacturer and also for finished product manufacturers. So the next question is it possible to apply for one CP for a substance produced at 2 manufacturing sites if the manufacturing is the same? It is described this in our policy for revisions and renewals, and also for the in our document for the sister files. So it is possible to have different manufacturing sites for the final substance if they are part of the same group. If they're not part of the same group, for transparency, and the sister file application should be submitted. Okay, then the CEP 2.0 requires also sites performing physical treatment. Can you clarify which physical treatment are to be considered? Is warehouse to be included? No, it's really the sites which are involved in the manufacturing activities related to obtain a specific grade, which is stated on the CEP. This might be milling, micronization. This is what I see most common examples, or sterilization sites. So the sites which are doing warehouse are not stated on the CEP, only for warehousing. Also the quality control sites or the sites which are involved in the stability testing are not mentioned on the CEP. Okay. Is there a need to keep the overall quality summary up to date during the life cycle of the CP? It is highly recommended for you to, and we have now a new document on the quality or overall quality summary, and its use in the evaluation procedure. So now it is requested at the submission of the new dossier application. But of course, if you introduce some major or important changes to the manufacturing process or the control strategy, it is highly recommended to keep it up to date. So I continue with the questions. Can marketing authorization holder use EP monograph method if the CP holder uses in-house method for impurities and related and residual solvents? So if the as I mentioned during my presentation, if an additional in-house method is appended to the CEP, and it applies more specifically for impurity for related substances, this means that EP method is not suitable to control the quality of this substance specifically, because this substance may contain additional impurities, in-house impurities, at levels above the reporting threshold, which cannot be controlled by the monograph method. So if the additional methods are appended to the CEP, this you can conclude that the EP monograph method is not suitable. There is something additional, and the additional methods should be used. Then it can be alternative methods which are used by the marketing authorization holder, which can control these impurities, but it's out of scope of CEP then, and it should be really the relationship between CP holder and the use of the CP, and it should be addressed in the marketing authorization application. So, can residual solvent tests be omitted from the specification, considering the results of commercially manufacturing batches? So, as it was mentioned in my presentation, the solvents which are used in the last steps of the manufacturing process should be controlled routinely in the final substance. Okay, for the solvents which are used prior to the final steps, based on the available results for commercial or validation batches, if they are detected below 10% of ICH Q3C limit, the control can be omitted. But the solvents which are used in the last step of the manufacturing process should be controlled because they are most likely to be present in the substance. If acetone and purified water are used in the last step of the process, is it necessary to include this control in the CP application? Yes, as I told that if they're used in the last step of the process, it's necessary to control them in the final substance. So the next question, is RMS is mandatory? No, the risk management summary for elemental impurities is not mandatory. It is recommended to submit it. So for more transparency on the CP regarding the elemental impurities which can be present in your substance, but even if it's not submitted, the CP will be transparent on the introduction of classified elements in the manufacturer by the corresponding statements on the CP. So, hello, you mentioned that if EP monograph is suitable to control the quality, the in-house method tests are not appended to the CEP. Can the in-house method tests still be used in such cases or not? Yes, they can be used, and we don't ask them to remove them from the CEP application. However, it is concluded that even if you used your in-house method, EP method is still, so it is equivalent to the monograph method, and the monograph method can still be used to control the quality of your substance. So you can still use your methods if they are still part of the CP of the CP dossier. So the next question is, how long the hybrid CP remains as hybrid CP if there are no changes in the CP procedure. Actually, now all the old CPs from September 2023 switch either to hybrid or to CP 2.0. So the hybrid CP may remain hybrid CP for many years. We do not force you to switch to CP 0.0. Yes, so the old CPs will disappear with the time with the revision. So as per when the CP is revised, it goes to hybrid CP if CP 2.0 was not requested, or CP 2.0. So the hybrid CP may remain. So today there is no request to switch to CP 0.0. So the hybrid CP may remain as long as possible. In case of sister file, for a different manufacturing process with a necessity to include particle size distribution, it is possible to include two subtitles. For example, one in order to distinguish a different manufacturing process, internal code, and one for particle size distribution. It's true that it becomes a little bit complicated, so it's not recommended. However, it's true that if the most important is that the impurity profile of the final substance is the same. Then the same substance can include tests for the subtitle for non-micronized and micronized material. It's true that if you include, in addition, the alternative manufacturing processes which are used to obtain these grades, it should be really clearly described in your CEP application, and it should be clear from the CEP dossier that the impurity profile of the substance is the same and which process is used for which for which grade. The next question. Will AMA spore and OMS IDs replace the actual address? As now both of them are stated. No, for the time being, the addresses of the manufacturing sites are still mentioned on the CEP, and they will be continued to be mentioned. If the decision is taken at one point to replace, there will be proper communication from the EDQM on this later. So then, can a manufacturer widen limits of a CEP already? Can a manufacturer widen the limits of a test already described in the CEP? Yes, it is possible if it is still in line with the respective guidelines. For example, in some cases, tighter limits are proposed initially for residual solvents or for some impurities, and it is possible, but it should be justified. And if it's in line with the respective guidelines, it is still acceptable. So the next question, in case where the heavy metal is stated under the new look of CEP, is it considered as mandatory test as the test is deleted under 248 from the monograph? It is true that most of the monographs were revised to delete the test for heavy metals if they are not if there is no risk of their presence. So this means that these tests are now not mandatory if there is no any specific risk for the presence. And if you include a test for heavy metal in your specification, most likely you will have to put these tests to the section for the tests which are non-required by EU in line with EU requirements. There is a separate table in the CP 2.0, which for the tests which are outside of EU requirements, because today we consider that if the compliance with ICH Q3D is demonstrated and that if the test is not foreseen by the monograph, it's not required. Then we have a question. If EP monograph requires identification testing for chloride following general chapter, however, DMF states that USP quantitative chloride test is being used instead. Does QC still required is still required to complete ID testing following EP monograph? It should be justified in your application that the test that you use for chlorides is also used for identification test, and in this case it will be considered as in-house test, and if it during the assessment it is demonstrated that this test is at least equivalent to the pharmacopoeial test, it can be accepted to have this test. However, the additional test will not be stated on the method will not be appended to the CEP, and for the users of the CEP, they can still use the test identification test of EP monograph for identification of chloride. We still have time. Yeah, so we stop here with the questions, and hopefully you enjoyed the presentation and it was useful for you, and it will allow you to be more yes, to be more clear in the CEP applications, and hopefully it will allow also to fasten the review assessment process and granting the CEPs for you. Anais, I give you some final words. Thank you for your attention. Have a good day.
Yes, thank you so much for attending this presentation this morning. We hope you enjoyed and that it was useful. Please, again, we have not been able to go through all the questions because we receive a really important number of questions. So if you have general questions, please check our FAQ on our website. If you don't find the answer, you can email us a general question on the help desk. If it's for a specific question related to your application, you can also contact us by email referring to the application number, and this is it. We wish you a very nice day, and goodbye from Strasbourg. Goodbye.
Goodbye.